Okay, so we're going to be talking about cell structure in this particular section, and I love talking about cells. They are just absolutely fascinating structure. So we're going to talk about that and really dig into it. So the key questions that we're going to cover in this particular section is really talking about all of the specific organelles, and we're going to group them together by their purpose, by their function, pretty much. So the cell nucleus kind of really stands on its own. It's the control center of the cell. Then we're going to talk about organelles that help make and transport proteins. So the proteins are so important. DNA actually codes for proteins, and then proteins are what actually carry out all of that information that is coded in DNA. We're going to talk about the functions of vacuoles, lysosomes, and cytoskeleton. Our cells actually have their own skeletal structure inside of them. Then we're going to talk about the functions of chloroplasts and mitochondria. We're going to just kind of briefly talk about them, but in the next couple chapters, we're really going to dig in and talk about the processes that occur inside of these. So chloroplast is where the process of photosynthesis occurs, and mitochondria is where the process of cellular respiration occurs. And so we're really going to dig into those processes, but we're going to do that at a later date. And then the last section we're going to talk about is the function of the cell membrane. And the cell membrane is that membrane that completely encompasses the cell. And it's what keeps that cell together. It's what keeps that cell safe. It's what keeps that cell protected. It's also what allows things in the cell and allows things out of the cell. So it is really, really important that we have a working and solid working, not like solid that there isn't any holes in it, because there definitely are. We're going to talk about all the things that allow that the cell membrane uses and functions. So the book, if you're following us along, we're on page 247, and I recommend following along because that way you can draw the sketches on our little flip cards that you have started already for me. You've already cut them up. You should have about 16 to start off with. Now we may have to add a few cards here and there. And I am going to mention throughout this video what your next card should be and should look like as we go through this, okay? So the book uses the cell and the analogy of that the cell is like a factory. You may also hear sometimes refer to the cell as like a city. So in this case, you're going to see this analogy a lot. And it just really helps you understand that there is a structure and there's a function and there's an organization to the cell. It isn't just this random circular blob that just happens to build organisms. There's a lot of things inside of these cells that make it work, and it's just absolutely fascinating. It's one of the main reasons that I one of the things that supports why I believe in a God, and I believe in the God, because when you take a look at all these incredible specialized things, it's like, how could this just happen by chance? There must be some kind of an amazing designer behind this. And so and that just, I think this section really solidifies that for me, so I really loved teaching that. So they're very, very busy. There are patterns that you can see once you understand what they are. Looking at this image now, it might just be really confusing and just kind of disorganized, but there really is an organization to this. We can start by splitting the cell into two main parts, the nucleus and the cytoplasm. The nucleus is this large section, usually found in the center of the cell. At least in animal cells and plant cells, you may often find it up against a wall, but for the most part, it's going to be in the center, and everything else is the cytoplasm. Cytoplasm, so here is your first card, cytoplasm. You're going to simply draw a cell on one of your flip book pages, and inside of it, you're just going to draw an arrow and label that cytoplasm. Cytoplasm, on the back of that card, you're going to write the facts about that specific organelle. And in this case, cytoplasm is the portion of the cell outside of the nucleus. So it's everything from the nucleus all the way out to the cell membrane. It is where all the organelles sit. The cytoplasm is like a jelly-like substance. It is like really watery jello or like jelly jam. Things can slide through it, but it still has this gelatinous mass to it. And cytoplasm really helps give structure and support to that to that cell. If there wasn't that cytoplasm, it would just be this empty space, and so the cell would not have the support that it needs. So that's your first card. Your second card is organelles. So you're going to label another card organelles, and you're simply going to draw a couple examples of organelles. If you don't know what any organelles look like right now, that's fine. You can draw them later as we learn them, or you can look in the book or on the slide there and pick a few of those images that you want to draw. So organelles, it literally means small organs, pretty much. So organ, you can see the word organ there, and the suffix "els" at the end of it, a word, usually means like a small version of it. So organelles. So organelles are literally small organs inside of a cell. They have very specific functions. They follow very specific instructions and produce very specific products. So your organelles are tiny organs within the cell that have very specific functions. So I said the book compares the cell to a factory. Again, if you've ever been to a factory or watched a video on a factory, you've seen things where it is very specific. The same machine that puts the doors on a car can't do anything else. That is its only job. If you've ever been to Krispy Kreme and you've seen them making the donuts, the part of the assembly line that drops the icing on the donuts, that is its only job. It can't do anything else. And that's how the organelles inside of a cell function. So the first cell we're going to look at is the nucleus. So this is your next card. This should be your third card. You're going to draw a cell, and inside of that cell, you're going to draw a dark circle. Again, the nucleus is the control center of the cell. It's like the main office of a cell where the CEO and the boss and the managers all work. So it is the control center. It's often referred to as the brain of the cell as well.
Attention, Tom, please call Nicole at extension 8929. Nicole at extension, 8629. Thank you.
Sorry, guys, I'm recording at school before I leave, and so there was just an announcement. I apologize for that. So the nucleus contains nearly all of the cell's DNA, and with it, the DNA is all the coded instructions for making the proteins and all of the other really important molecules that we have in our cells. So the nucleus holds all of those instructions. That is where you're going to find the instruction manual for your body is going to be inside of that nucleus in the form of DNA. The nucleus is surrounded by what we call a nuclear envelope or a nuclear membrane. It's a 2-layered membrane, and it is filled with holes. We call these nuclear pores. Just like our skin has pores that allow things to go in and out of our body, specifically out, like we sweat through our pores. These nuclear pores allow things to come in and out of our our nucleus. Things that need to go in and out is things like mRNA go in, reads the DNA, comes out. Ribosomes come out. Various other things need to be able to go in and out of the nuclear membrane. The nucleus also holds our chromosomes. Humans, we have 23 pairs of chromosomes inside of every single one of our cells. And most of the time they are found in the form of what's called chromatin. It's more of these like thread-like, so they're more like yarn or string. They're sometimes often hard to see. But when the cell is ready for something called mitosis, which you learned about in 8th grade and we'll learn about in more depth here in a few months, they will condense into chromatids and form these chromosomes. And then that's when you can actually see them inside of our cells. So if you think back to the onion cells that we saw the other day, you may have actually gotten to see some of the chromosomes because they were in a condensed form. The next card you need is the nucleolus. So you're going to need to draw a cell with a nucleus, and inside the nucleus, a dark circle called the nucleolus. And the nucleolus is where ribosomes are created. So it is found inside the nucleus, and ribosomes are formed inside of the nucleolus. So the next chunk of organelles we're going to talk about are in the process of building proteins. Proteins are so incredibly important. It is literally, our DNA codes for proteins. Proteins are then what carry out all the functions of the body. So that is what is happening during that is what is happening here. So we will dig into protein synthesis a little bit later. It is a long, complicated, can be complicated process. I can definitely break it down for you, and we will do that at a later date. Okay, so a lot of the structures inside of a cell are simply involved in this process, the production of proteins, and then also the production of some lipids and carbohydrates. So a lot of our organelles are making and distributing our macromolecules. So the first one we're going to talk about, you guys will need to make another card. These are called ribosomes, okay? And these are everywhere in our cells. They are one of the most important jobs is that they're their own tiny little factory. And they are what actually make the proteins. And so these guys are really, really important. So mRNA has read the code of the instruction manual of DNA inside the nucleus. It carries it outside the nucleus and it goes to these ribosomes. And then the ribosomes read the DNA. They read these codes and they read the instructions and they form the proteins. They read the DNA in chunks of 3. And then that chunk of 3 bases from our DNA actually tell the ribosome what amino acid to grab. And then transfer RNA, which is also hanging out in your cytoplasm, then brings that amino acid kind of over. And then it creates this long chain, and long chains of amino acids are proteins. So this is where our proteins are really, really made. So it's really, really important. They're like their own little mini factory inside of our larger factory. So for your card, ribosomes are really, really tiny little dots, and they are found on a lot of things. So you're just going to draw, you can draw a couple just small circles, small kind of dots, and you're going to see these throughout the cell, literally hanging out loosely in the cell, but you're also going to see them attached to other organelles in here. Okay, so on the back of your ribosome, you want to write that they synthesize proteins. Remember, synthesize is a fancy term for to make. And they are found on rough ER, and they're found in mitochondria, and they are found floating around in the cytoplasm. Okay, so they are the one that are actually creating the proteins that the DNA inside of our nucleus have coded for. We have a lot of them, especially in cells that are very, very active in protein synthesis. So cells that need to create a lot of proteins are going to have a lot of ribosomes. So here's a fun word, endoplasmic reticulum. We often refer to this as the ER so that you don't have to write endoplasmic reticulum every single time. And endoplasmic reticulum is where lipids are synthesized. So this is where lipids are made. Remember, lipids are those fatty acids. They also are proteins and other materials are exported from the cell. So endoplasmic reticulum is in charge of making lipids and then also shipping out proteins and some other cells. So there are 2 pieces of endoplasmic reticulum. We have what's called the rough ER and we have what's called the smooth ER. And the main difference between them is that rough ER is covered with ribosomes. So the rough ER portion of ER is involved in the synthesis of proteins because the ribosomes are found on it. So the ribosomes make the proteins and then they pretty much leave the ribosome, they enter the rough ER, and then they can be modified. So if they need to be adjusted at all, then that is when the ER is going to adjust them. And then the ER, once they are all perfected as they can be, the ER is what then sends those proteins out of the cell. Okay, so they are secreted from the cell, a lot of membrane proteins. There's proteins destined for just other locations within the cell. So the rough ER is responsible for getting those proteins where they need to go, whether it's somewhere in the cell or even out of the cell. Again, rough ER is going to be very abundant in cells that produce a lot of proteins that are meant to leave the cell. Okay. Other cellular proteins are made on free ribosomes. So ribosomes that proteins that might need to stay in the cell could be made on proteins that are just kind of hanging out freely in the cytoplasm. Proteins that need to be sent out of the cell are going to be made more on the ribosomes that are attached to this rough ER. Then the portion of ER that's known as smooth ER has no ribosome. So that's smooth because it's not covered with all these little tiny dots. And the smooth ER contains this collection of different enzymes that really perform these really specialized tasks. So think back to when we learned about enzymes and how they can speed up reactions and stuff. And one of these things that the smooth ER does is it also creates our lipids. And it really helps in the detoxification of some different drugs that we tend to put into our bodies, good and bad drugs. But this is where this happens, is it's the detoxification. And it also plays a role in the creation of carbohydrates. So you're going to make two cards here. You're going to make a smooth ER card and you're going to make a rough ER card. The structure of ER is going to look similar to each other, and they are going to look, and remember, I have an example over there by my desk, so if you need to see what I'm talking about, I pretty much make a big kind of like, if you can see my mouse, I just kind of go like that on the slide. So again, a lot of like a big scribble back and forth. And then for rough ER, I make lots of dots on that. And then the smooth ER, I just leave off the dots. And again, on the back of those, you're going to talk about that the rough ER is kind of like the cell highway. It's how those proteins travel around. It transports a lot of those protein specifically out of the cell. Smooth ER is more involved in the protein, the synthesis of lipids and carbohydrates. Our next one, so here's that rough ER. You see what I mean by like I just kind of scribbled back and forth. So our Golgi apparatus is also referred to as a Golgi body. I have always referred to it as a Golgi apparatus. That's what I learned it as in high school, and then in college, that's what I still called too. But if you ever see a Golgi body or a Golgi apparatus, it really is the same thing for the most part. The reason I prefer to call it Golgi apparatus is because that's the big chunk. Sometimes you'll have these little tiny vesicles that are actually pieces of the Golgi membrane that has broken off to move something around. Those are often also referred to as a Golgi body because they are still a body made out of the Golgi membrane. And I feel like that sometimes is confusing because you're not quite sure what I'm actually talking about. So I refer to it as the Golgi apparatus. But do know that it also has the name of the Golgi body. So if you ever see that, you're like, what is that thing? Mrs. Stewart didn't teach me about that. It's the same thing. And Golgi is named after somebody. And I draw this as a couple like flat pancakes. You see how those are kind of individual and they're flat. So this is another one of your drawings. So Golgi body, Golgi apparatus, and again, kind of some flat pancakes with little to no space in between them. This is still involved in the production and the movement of proteins. So proteins produced in the rough ER then move to here. It looks like it is literally a stack of flat membranes. So as proteins leave the rough ER, they have these, what they kind of refer to as an address tag that tells the Golgi body where they're supposed to be going. Okay, so the Golgi body then reads those address tags, and then they are bundled into these little structures like this. So let's say that this is a protein here, and they are moved from the endoplasmic reticulum, and then the Golgi body gets it, and it kind of moves through this, and again, the tags are read, and then they are put into the Golgi body, or Golgi apparatus literally kind of breaks off part of its membrane, envelops that protein, and then sends it wherever it's supposed to be. So think of the Golgi apparatus as kind of like the UPS of the cell. They're what's actually shipping the stuff around. So it modifies, it sorts, and it packages, and then the stuff from the endoplasmic reticulum for storage in the cell or release from the cell. So it is really sorting it like the UPS would, sorts it, packages it, and so on. So this is going to be, again, if you haven't done already, this is another one of your cards, Golgi apparatus or Golgi body. You can write both of them on there. Looks like flat pancakes. And they they said they modify and package and ship off pieces of the proteins that are made. Okay, so those are all of the organelles that are that are grouped together in the function of proteins. So now we're going to move on to organelles that store, clean up, and support our cell. So we're going to have things called vacuoles and vesicles. So a vacuole, here's another card. You're going to create a vacuole, and a vacuole is just a really large sac. It's like almost like a balloon inside of the cell. So you're going to just kind of draw, in this case, I would draw a rectangular box, and then a vacuole is a big circle that almost fills that whole rectangular box. And that is because vacuoles are these large membrane enclosed structures that are mostly found in plant cells. We don't have these large vacuoles in our bodies. And the vacuoles store things like water and salt, proteins, carbohydrates. So in those plant cells that we looked at earlier or last week, you notice that the nucleus was kind of smushed up against the side. And in this case, it's that way because the vacuole is taking up almost all of the space. It's holding all of that water for later use. It is a storage facility. So when a plant gets wrinkled and shriveled up, it's because the vacuole in there has shrunk because it doesn't have any water left in it. And so that is what this is for. So this is going to be your next card, the vacuoles, and they store materials. So again, you want to draw and I have you draw a rectangular shape because cells, plant cells look more like bricks in a wall. They're very uniform in their shape. Okay. Most plants have this really large vacuole. It really does help in the rigidity of the cell and the rigidity of the plant. So the vacuoles are the big ones that we find in plant cells. Now your next card is going to be called a vesicle. Okay, so now we're going to be talking about vesicles. And most eukaryotic cells are going to have something called vesicles. And vesicles are kind of small versions of vacuoles. Vesicles are going to be a lot of different things. So they're just a small, so again, draw in kind of a cell, and then these are going to be small. They're going to be bigger than ribosomes, but they are membrane-bound sacs, and they are going to do numerous things depending on where they came from. So some of these vesicles are going to, what you saw with that Golgi apparatus, how the Golgi membrane kind of wrapped around the protein and moved it around. That can be referred to as a vesicle because it is a small membrane-bound organelle in there, and it is moving something around. If something bad gets into a cell, the cell membrane may wrap part of its cell membrane, like break it off and wrap it around something bad and get it back out of the cell. If there's a cell that is dying or a structure inside of a cell that's not doing well, a vesicle, a specific one we'll talk about in a minute, is going to wrap around that dying or broken organelle, and it's going to digest it and break it down. So vesicles are just these small membrane-bound organelles inside of our cells, and they can do a variety of things: store materials, move materials between the different organelles, send stuff to the surface, bring stuff in from the surface. So there's a lot of different roles that a vesicle can have. This one in specific is called a lysosome, so it is another small vacuole, and it is filled with enzymes that are there to break down stuff. This is our cleanup crew, kind of like the janitors of the factory. So they break down lipids, carbohydrates, proteins back into small molecules that can be redistributed throughout the cell and reused. They also, like I said, break down organelles that are kind of getting old or outdated or really aren't doing their job. It's going to remove junk that's in that cell, and it's going to just, like I said, break it down into smaller pieces and then redistribute those pieces back out into the cell so that it can be recycled and reused. There's a number of really rare but serious human diseases that can actually be traced back to lysosomes that fail to function properly. So these lysosomes are not getting rid of junk out of the cells. These lysosomes are not breaking down old junky cell pieces, and those can create some diseases. Okay. So that's it. That's the vacuole and vesicles. That's the organelles that store, clean up, and support. So sorry, make sure you make a card of the lysosome. Again, it's going to be a small, round looking organelle. And you are going to, again, make sure that you talk about that it gets rid of waste, that it breaks down, old damaged cells, damaged cell pieces, and so on. So now, sorry, let me go back. This is organelles that capture and release energy. So I told you that our cytoplasm has what we call a skeletal structure to it, and we call this the cytoskeleton. So go ahead and make another card, and you're going to label this one cytoskeleton. And it's exactly what it sounds like. It is the skeleton within the cell, the cytoplasm. And it's a network of what we call protein filaments. We're going to talk about those in a second, that really give these eukaryotic cells their shape and their internal organization. So think of it as like the walls inside of a building. It breaks up the building into rooms. The cytoskeleton kind of does the same thing. It helps kind of maintain its shape, and it also helps things move. So it also is the hallways, the doorways, things like that. So under the cytoskeleton, you're going to put things like microfilaments and microtubules. Okay, so microtubules, you're going to draw them as I think they look like little tubes, okay? And the microfilaments, again, are like thread-like structures. So you're going to draw these thread-like structures and these little tubes, and you're going to make sure that you have them labeled microfilaments and microtubules under the one that we call cytoskeleton, okay? The tubules are hollow structures, and they are really important in cell division. Okay. Then there's something called centrioles. So you want to draw these. All of these things are going to be under cytoskeleton. Then you have these little centrioles, and they kind of look like two marshmallows, is kind of how mine look when I look at them. So two marshmallows, and they're right next to each other. Actually, they kind of form a V so that the two inside corners kind of touch and they kind of V out. And these are really important in mitosis, so they're going to be located near the nucleus. Okay, they are not found in plant cells. They're only going to be found in animal cells, but they are helping in cell division. Okay, so then we have our chloroplasts. So chloroplasts is what helps actually take in energy. So you're going to need another card for this. So this is your chloroplast. Sorry, I'm trying to find my card so I can make sure I tell you how to do it and draw it properly. Chloroplasts are kind of an oval-like shape, and they have these flat coin-like stacks inside of them. They have a really fun special name. We'll talk about those when we learn about photosynthesis, but it's a thylakoid. And so in our factory, these would be the solar panels on our roof. These are what's collecting energy specifically from the sun and is converting that energy into chemical, into food during the process of photosynthesis. So again, you're going to draw an oval shape with like stacked coins like that, just in this image. And they are capture energy from sunlight and converted into chemical energy, specifically stored as food in photosynthesis. There are two membranes that surround the chloroplast, and inside of these membranes are these stacks. And inside of these thylakoids is the pigment chlorophyll. Chlorophyll is what actually makes plants green. There are other pigments in there. Green pigment is just the one that is kind of the most abundant. And so we will talk about the different steps of the process of photosynthesis at a later date. So make sure, again, you have a chloroplast. They are green, they are oval shaped, they contain chlorophyll, they are only in plants, and again, they convert sunlight into food sources. Okay, then we have the mighty mitochondria, and it is the powerhouse of the cell. It is where, so photosynthesis creates glucose, that's C6H12O6 molecule, also creates, so it creates that in water and stuff. Not water, sorry, said that wrong. And oxygen. And the mitochondria then takes that glucose molecule and converts it into what's called ATP, adenosine triphosphate. And ATP is what actually is the energy that cells use. Now, plant cells do have mitochondria in them. Plant cells do go through cellular respiration. Often that is a misconception that students think that cells only do photosynthesis and plants only, and then humans or animals only do cellular respiration. That is false. We can't do photosynthesis, but plants do both. So plants do photosynthesis and cellular respiration because they use that cellular respiration. They use that to create the energy so that they can do their own things. So mitochondria converts that chemical energy stored in food molecules like glucose and turn it into compounds that are more convenient for the cell to use. Mitochondria also has two membranes. And in humans, nearly all of our mitochondria originate from the cytoplasm of the mom's egg. So all the mitochondria that you have in your body duplicate itself from the egg cell from your mom, because when the sperm comes and fertilizes the egg cell, a lot of that sperm is broken down, that it just kind of gets to the DNA and the chromosomes joined together, and then mitosis occurs from there. So most of the mitochondria that you have comes from the cytoplasm in your mom, from your mom. There is some science. Again, this is just science. This isn't necessarily anything you have to believe. It's not really stated in the Bible, obviously, but that sometimes mitochondria was actually, because it looks very similar to a bacteria. If you really look at it, similar in size, similar in shape and design to a bacteria. And so it was thought that at one point in time, mitochondria was its own living thing. And then it got into a cell, and the cell was like, hey, this thing's really beneficial. This is helping me get energy. And then it became the symbiotic relationship. This endosymbiotic theory is what it's called. And then they now are a part of our cell, but it is said that maybe once upon a time they were their own thing, because mitochondria has their own DNA, which is why it said, when we learned about the nucleus, it says nearly all of our DNA is in our nucleus, and that's because some DNA is found in our mitochondria. So our mitochondria is another cell, so make sure you have that. Again, it's like this bean shape, and it has this outside kind of membrane, and then this membrane in here. And there again, this is where cellular respiration takes place, and it's a really, really cool but somewhat complex process we're going to learn about in a different chapter. So on this page, you're going to write mitochondria. Again, kind of draw it like a oblong bean shape. And you're going to talk about the fact that it is the powerhouse of the cell, is where ATP is made, and so on. Okay, so really important organelle there. Okay, now we have our boundaries, our cellular boundaries. So this is the stuff that kind of surrounds our cells. So cells are surrounded by a barrier known as cell membrane. There are also cells that have what's called cell walls on them. Many cells, including most prokaryotes, produce this really strong supporting layer that surrounds the membrane, and that's called a cell wall. That is what helps make plants very rigid. Because it is a cell wall. So think of a wall. So think about, would you want to live in a building made out of a bouncy house, like a big bouncy house building, or would you rather live in a brick building? So that's kind of the difference. The bouncy house building does have structure and it does have support, but think about how much stronger and how much bigger you could build a brick building or a skyscraper. You couldn't really have a skyscraper made out of a bouncy house the size of a skyscraper. It's not sturdy enough, and that is because we don't have cell walls. That's why humans don't get as tall as buildings. Our cells don't have the support to hold, stack each other up like that. So you're going to make a card for cell walls. The main support here, the main function is for support and shape, and it protects the cells. So in this case, you're just going to draw a rectangular shape, and right inside of that, you're going to want to make a small kind of, almost like it's kind of like the vacuole one that we did, but you want to make that small oval kind of fill in almost right inside the cell wall, because you can do the cell wall and the cell membrane in the same card. And then you can also do cell membrane by itself. But I do want you to know that the cell membrane is on the inside of the cell wall for organisms that have both. And then organisms that just have a cell membrane, that's all that they have. So it's up to you if you want to do one or two cards. You can combine them. I would just have two images. I would have the cell wall with the cell membrane together, and then another image with just the cell membrane. I don't want you guys to think that the cell membrane always goes with the cell wall. It doesn't, okay? Both of these are porous, okay? We're going to talk about the cell wall first. It allows things like water and oxygen, carbon dioxide, other substances to pass through it. Because remember, our cells, the chloroplast has our cell, is inside of that. Mitochondria is inside of our cell. In order for our cells to get, for us, we don't have chloroplast. So we have to get our glucose from the food that we eat. So the glucose has to be able to get into our cells. But again, the cell wall allows for things like that to move in, and again, provides strength for plants. So then we have our cell membranes. Yes, cell membranes look that complicated. They are phospholipid. It's also called a lipid bilayer. We talked about this when we talked about lipids at the beginning of the year. There's a hydrophilic and a hydrophobic portion of our cell membrane there. All cells contain cell membranes, and they are generally made of this double layered sheet called a lipid bilayer. Okay, the parts on the inside are hydrophobic. They're afraid of water. The parts on the outside are hydrophilic, and they like water. Okay, so you're going to, again, in that cell membrane card, if you made a separate one or a single one, you want to talk about that there is a bilayer. And again, the cell membrane, just like the cell wall, regulates what comes and goes inside of our cell. It also does give some protection and support to our cell. So we need to know a little bit about lipids. The layered structure of cell membranes reflects the chemical properties of that lipid. Remember, we talked about that there were things called hydrophobic, which is water hating. If you hear the word phobic, and phobias means that they're kind of afraid of. So it's a way to think that it doesn't like water. And hydrophilic means that it loves water. And so the hydrophobic parts are on the inside of the wall, and the hydrophilic parts are on the outside of the wall. And this is going to play into when we learn about all the processes that are entailed in things going in and out of our cell. Our cell walls are what's called selectively permeable, which means that it has holes. So think of like a colander or a strainer that you might use. Let's say you made pasta and you have to dump it into the colander because you're trying to get the water out, but the noodles don't come out unless you have a colander with really big holes. So again, you have to adjust the size of your colander so that it doesn't drop things. And that's what a membrane is like. It is semi-permeable, which means that some things can get through it, but other things can't. So in this image, you can see that the small green and purple and yellow dots can go through, but the bigger blue and the big red, which ones happen to look like red blood cells, cannot get through. They are too big for the holes. But there are processes that will allow the cell membrane to let those bigger things get into the cell. And we will talk about that when we talk about the different functions of the cell membranes. And we have this thing called a fluid mosaic model. So proteins molecules are often embedded inside of that lipid bilayer of almost all of our cell membranes. Okay, and then carbohydrate molecules are then attached to a lot of these proteins. These proteins move around and they can float, and so a lot of these molecules that are found in the cell membrane, scientists refer to them as fluid mosaic because they are moving around. Okay, so fluid means, like, think of like a fluid. It can move. You can slosh it around in a jug, you can shake it up and it moves, so it's very fluid. And so some of these proteins form these channels and these pumps that help materials move across that membrane. So like I had said, that there were processes, and we have these processes that allow bigger things in and out. And it's because these proteins and these carbohydrates have formed these channels, these pumps that will allow these bigger things to move through them. The carbohydrate molecules actually act like identification cards, so some ID cards that allow cells to identify each other. And then some proteins actually attach directly to the cytoskeleton themselves, which then allows the cell to kind of respond to their environment by using their membranes to help move or change their shape. So our cells move a little bit because if they didn't, you wouldn't be able to move. So as you move, your cells have to kind of slide around each other, and they have to receive communication from things like nerves, the neurons, that says, hey, I need you to move because this person's going to be walking, or so on. And so there is a lot of stuff going on inside of these cells. So that's it for right now. I know that was a lot. We will spend some time going over we've got some activities, some online activities that we will be doing. I do have potentially some labs or whatnot. We've already built a model of the cell once, but we'll probably build a more in-depth cell where you guys are actually going to get like Ziploc bags, and you're going to create a more complicated version of what you guys did last week. You guys made a very basic and eukaryotic cell, but I do want you guys to potentially make a more complicated version of the cell. So again, I have an example. Mine is not exactly the same as yours. I've used mine for years, and so, but it does have the same kind of gist of what you guys are doing. So if you need to see that, it is hanging there next to my desk. If you have any questions, please feel free to email me, ask me any of the questions. The quiz will most likely be Thursday to Friday, really depending on how long it takes you guys to get these notes and so on. Okay, so I hope you guys have a great day.